Good morning. You awake now? Kind of. Good morning. Good morning. It's better. I know it's a nice day and you'd rather not be awake or something. I don't know. Whatever. Today we have to go through this chapter. Chapter 27. Right? You can get that. Whatever. You'll figure it out. Okay? So, have to go through this chapter and get it done today. This is all stuff that you have gone over in lab. Okay? So this should mainly be a review. This is a survey of animal diversity. All right? So we're going to be looking at eukaryotes and basically when they came about. All right? The idea in terms of evolution is that at some point eukaryotes evolved from prokaryotic cells. Okay? Nick? All right, so really we're talking about the animal kingdom, and namely multicellular organisms. So, we know animals are multicellular, heterotrophic eukaryotes, right, with tissues that develop from embryonic layers. And this week in lab, you'll be doing tissues. Okay? Muscle, nervous, epithelial, and connective tissues. You'll be learning about their characteristics and so on, right? And be able to identify them in under the scope, which will serve you well to start next semester, next year in anatomy and physiology. All right? So, there are exceptions to this, but really we're going to talk about what characteristics of animals and multicellular organisms we use for classification, right? Because that's what you basically did in lab, was classify, figure out dichotomous keys, what characteristics, why is one animal this, if not that, etc. So, cell structure. All right, because they are multicellular, cells began to specialize. All right? So, cells begin to specialize, they lack cell walls. Right, your bodies are held together by proteins such as collagen, that is connective tissue. All right? And then you have nervous and muscle tissue, which are unique, defining characteristics of animals. What is unique about nervous and muscle tissue? Jason and Rob. It has a protective covering over it. All right, it has a protective covering over it. What about muscle and nervous is special? Sarah?
Involuntary? Well, nervous is and muscle can be both.
Okay. Involuntary in terms of what? What happens to those two tissues? They do work together. What can happen to those two tissues?
They can carry an electrical impulse.
They carry electrical impulse. Thank you, Nick. Thank you. I did not fail as a teacher for the most part, right? Carry electrical impulses. They are excitable. Okay? That is not, right? So you can excite a nerve to relay electrical impulse. You can excite a muscle for contraction. Okay? And so tissues are groups of cells that have common structure, function, or both. So it's cells grouped together to do a specific function. All right? Or structure. And usually it's based upon function. All right? Reproduction in terms of multicellular organisms. All right? Most animals reproduce sexually. All right? There are a few exceptions. All right? With the diploid stage being the dominant cycle. Sperm, egg come together, two haploid cells form zygote, a diploid zygote, which then undergoes rapid mitotic divisions known as cleavage. Okay? So when I showed you that frog embryo development, right? Where we started, that first separation, you ended up seeing a cleavage furrow. Okay? It's sort of beginning of an infolding of a mitotic division. Then, very quickly, it went from 2 to 4 cells to 8 cells very quickly. Those first divisions are what we call cleavage. All right, very rapid mitotic divisions. All right, then cleavage, the idea is that dividing cells produce more cells. Cleavage produces leads to the formation of a multicellular hollow blastula. Right, so that ball of cells sort of grows and is actually hollow. There's fluid that goes inside, and then what you see is a ball of cells, a sphere of cells with a hollow inside. That is a blastula. Okay? And then at one point, the blastula undergoes a process of cell movement, right? Whereby some cells that are on the outside go to the inside, right? And the covering of cells move in. Okay, that is called gastrulation, forming a gastrula. Those cells that move in go through what's called the blastopore. Okay? Does the blastopore, that is a familiar word for you, yes? Yes? All right. All right. Eventually, the point of this is that there are now different layers of cells forming your ectoderm, endoderm, and maybe mesoderm. Okay? Here it is in sort of graphical format. Your zygote, spermatic fusion, 8-cell stage, you have cleavage, grows up, ends up into this formation. Now you'll see that there is a certain way that cleavage occurs. Okay? And depending upon the type of animal, the characteristics of that animal, cleavage can be what we call radial or spherical. You'll see this in a little bit. Eventually leading to the blastula, all right, and then you end up cut it in half, right, through this sort of section. You end up, you can see the inside. It's a hollow space. That space in the middle is called the blastocele. And then you begin to see the beginnings of gastrulation, where cells move in and forming different cell layers. All right? And the beginning of that infold is the beginning of, what is that called? That's called the archenteron. What is that important for? What does that end up becoming? The digestive system. Okay, so what you see, that archenteron, that infold, that in that formation of that sort of infold area, which will eventually form a tube, is the beginning of the digestive system. Okay? And depending upon whether or not it's a deuterostome or protostome, that may be the beginning of the mouth or the anus. All right? Many animals have at least one larval stage. A larva is essentially immature and morphologically distinct from the adult. All right? Examples of a larva. What type of animals have a larval stage? Mainly Drosophila, insects, arthropods. Right? It eventually undergoes metamorphosis to the adult. All right? And then your juvenile resembles adult, but it's not sexually mature. So a juvenile is not a larva. Right? A larva is not a juvenile. There's two separate terms. Okay? Most animals, but not all, and only animals though, have what we call Hox genes. We went over Hox genes, correct? No? We didn't mention Hox genes at all? At all when we talked about Drosophila? No? We didn't talk about Hox genes? Yes, we did. We mentioned Hox genes. Okay? They're important in body segmentation. Okay? And patterns. They help regulate the body form. Right? So that you know where. How many vertebrae do humans have? Nick, what types of vertebrae are there? Thoracic, cervical, lumbar, okay, and sacral. Okay? How many cervical vertebrae are there? 7. 7. C7, T12, there's 12 thoracic, and L5, lumbar 5. Okay? So humans have 7. How many do gorilla have? 7. How many do cows have? 7. How many do giraffes have? 7. Okay? Why is that? Hox genes. Okay? So that certain mammals, okay, have 7 vertebrae based upon, because they share during their body pattern formation. Hox genes are formed and they sort of develop a certain way and distinguish where those vertebrae will be. And the fact that they have 7 cervical vertebrae. Okay? Now, giraffes' vertebrae are like that big each, as opposed to cervical vertebrae are about that thick in humans. Okay? They are conserved, but it can produce a wide variety of animal morphology. Giraffe vertebrae versus human vertebrae. All right? But they're mammals and they share Hox genes. All right? So, that's the idea of, we're going to go through the other stuff and a little characteristics in a little bit. A little bit of background. The origin of animals and multicellular animals is more than 500 billion years old. All right, so we span that sort of development from single cell to what we are now over 500 billion years. The idea was that we had a common ancestor somewhere between, in the rough area of 675 to 800 million years ago. You know, give or take 10 or 15 million. Just saying. It's kind of a wide range, right? It may have resembled what we call present-day choanoflagellates, protists that are the closest living relatives of animals. All right, so choanoflagellates, single-cell organisms, but they can live in colonies. Right? They have a flagellum, and they are somewhat similar to what you see in some of the cells of sponges. Right? So here's a choanoflagellate, and here's a cell of the sponge, a collar cell or a choanocyte. They sort of resemble one another, do they not? All right. But the difference being is that the choanoflagellate mainly lives a lot of its lifespan as a single cell organism. Okay? Whereby the collar cell, the choanocyte, actually resides within the sort of opening of the sponge. Okay? And then we have other animals that are more highly developed. All right. So a couple different years, a couple different eras we're going to talk about. The Neoproterozoic era, right, about 1 billion to 500 million years ago. This is beginning of sort of where our ancestry would be. All right, it dates from 565 to 500 million years ago. And we have lots of fossils that represent the beginning of animals. All right, so most of what we know about evolution of animals and multicellular animals comes from what? The fossil record. Okay? On the fossil record. Next era, the Paleozoic era. This is important because there is an event within the Paleozoic era called the Cambrian explosion. All right? This marks the earliest fossil appearance of many major groups of living organisms. Right? It was a time where a lot of different things occurred. All right? So it really was an explosion of diversity at that point. Okay? And you had new predator-prey relationships. One of the big reasons for the explosion of diversity was this: rise in atmospheric oxygen. Prior to this, atmosphere was mainly methane, other stuff. Okay, not much oxygen, so it wasn't being utilized by, well, could not be utilized by cells at that point. More oxygen, cells can adapt to that. All right, and then you have this is the beginning of the evolution of the Hox genes complex. Now animals are developed along that time. So Paleozoic era, Cambrian explosion. Then you end up with this fun idea or this fun graphical diagram showing you different types of animals that were present. Right? You look, there's a sponge, There's this whatever thing it is. Some worms are present. A lot of diversity came about in the Cambrian explosion. So then that continued through the Paleozoic era, but there were mass extinctions. So mass extinctions meaning that several different species would die off because they weren't adaptable to the environment. All right, and then eventually animals began to make impact out of the water and come out onto land about 460 million years ago. And then vertebrates made the transition to land around 360 million years ago. Okay, so out of the pool, per se. Then we get to the Mesozoic era. You have emergence of coral reefs. Okay, the ancestors of plesiosaurs. What are plesiosaurs? All right, so we're reptiles that return to water. So dinosaurs were the dominant terrestrial animal, all right, vertebrate. This is the beginning, the Mesozoic era, the beginning of mammals. Mammals are emerged, eventually flowering plants and insects diversified, and at this point, you end up mammal over. In the Cenozoic era, mass extinctions of both terrestrial and marine animals, namely due to the lack of resources. Mammals increased in size and exploited the vacated niches. All right, so dinosaurs die off, mammals sort of take over. Okay, and the global climate cooled, which allowed that to happen. All right. Questions on those eras. So you should sort of, I'm not going to ask you how many million years ago was the Cenozoic era? Or between 500 million and blah, blah, blah era, of blah, blah, blah million years ago, which era am I talking about? No. You should know that the Cambrian explosion happened when? That mammals emerged within the Cenozoic era or Mesozoic era, whatever. You should know general characteristics of each of those eras. Okay? Questions on that? So really that's sort of the background, where animals diversified from and what drove their diversity. All right? The next part of the chapter, and this says 32:13 because it's related to chapter 32 of the other book, and I like these slides better, so I use them. This is really chapter 27 in your book. Is body plans. Okay? And really, when we talk about a body plan, we're talking about a set of morphological and developmental traits. All right? What does an animal look like in terms of its shape and structure, and how did it get there? How did it develop those traits? All right? So, here are your early stages of development. Okay, what are we looking at? Zygote that has undergone what? In the step one, it's undergone what to this point? Cleavage. Right? So you're looking at an early stage, and by 32 cell stage, now something has changed. Look at them. There's a movement of those cells. Okay? Now it has undergone gastrulation. Right? And so there's your early gastrulous stage, site of gastrulation. You can see sort of the indentation. That's the blastopore. All right, and then, but if you block a certain transcription factor called beta-catenin with an antibody, what happens? So what you're looking at here is beta-catenin expression up here. Fluorescent tag beta-catenin. But when you block, and this is what it looks like when you block it. It doesn't undergo gastrulation. Okay? So an example of this showing that a transcription factor, a gene, is important in driving this process. Okay? And those genes that are regulated by beta-catenin are important in this process itself. Okay? Body plans involve symmetry. and developmental traits. Right? Structure, and the first rule of structure is symmetry. Types of symmetry that we're going to be concerned with. Julia. Radial. That was a good inflection. Radial. What else? Bilateral. What is a characteristic or asymmetrical? Right? What's characteristic of radial symmetry?
You can cut it anyway and it looks the same.
Okay, give me, all right, so cut it anyway and it looks the same, and then it's going to look like sort of the spokes off a wheel, right? So center and then radiates from there. All right. No front, back, left to right. Symmetry.
Split it in half, exactly the same.
All right, so you split it down one you split it down one axis and you end up with a mirror image. Okay? Most bilaterians have a development of a head, or what's called cephalization. Right? You see the arthropod lobster up there, head of animals. All right, there's a cephalization that occurs. But it's the left and right, and you show the difference between radial symmetry and bilaterians. Right? If you cut a bilaterian in half the wrong way, you end up with two, a top half and a bottom half. Okay? So you have a dorsal and a ventral side in terms of bilaterians, a right and left side, which are mirror images of one another, right? An anterior head or posterior tail. Right? So this is really the formation of the body axis, and then you have cephalization, the development of a head. So an earthworm, what would that type of symmetry would it be? Bilateral, right? It does, even though very primitive, it does have a development of a head region, right? With proboscis, etc. Okay. And radial animals are often sessile or planktonic, meaning they are drifting or weakly swimming. What does sessile mean? Sessile means stationary. All right. While bilaterians are mainly more active, they both have a central nervous system. Okay. Tissues. What type of tissues are produced when an embryo undergoes gastrulation? Brittany. Yeah. Those type of, yes. What type of tissues are produced when an embryo undergoes gastrulation? Ectoderm and endoderm. Okay? Ectoderm and endoderm. And most animals, all animals have at least those two, right? And then what's the third? The mesoderm, all right, or mesoderm. So those are your what we call germ layers. They give rise to your tissues of nervous, muscle, connective, and which one did I forget? Epithelial tissue. All right. So your ectoderm is a germ layer covering the embryo's surface. All right. So during development, you're going to have this germ layer on the outside, ectoderm. All right. And then as cells move in, they're going to produce two germ layers, and that cells that are on the inside are called endoderm. Okay? Most of your skin is ectoderm in germ layer origin, whereas your digestive system is mainly endoderm in germ layer origin. Okay? And everything in between is made mostly meso. Simplistic way of explaining that. All right, really, we're talking about diploblastic 2 germ layers versus triploblastic 3 germ layer tissue or 3 germ layer having animals. All right, so and these include your flatworms, arthropods, vertebrates, and others are triploblastic, whereas cnidarians and comb jellies are diploblastic, as examples. All right. Body cavities. What type of what animals have body cavities, and what's a body cavity? What's another word that you know for body cavity? Coelom. All right, so what do you call animals that do not have a body cavity? Acoelomates. Animals that do have a true body cavity. And which the one in between? All right, so pseudocoelomates. So you have a coelom, a coelomate. So here's your coelom, right? It has a separation, a body cavity in between. And what you see in blue, yellow, and red there are the tissue layers, right? And we're going to carry that over in terms of color. Example being your earthworm as a coelom. Okay? The pseudocoelom is a body cavity derived from the mesoderm and endoderm. Right? Your triploblastic animals that possess a pseudocoelomate are called pseudocoelomates. Okay? Look at the difference between the pseudocoelom. Right? There's complete separation as opposed to everything that's defined inside the mesoderm. All right? So separation between the two. Your acoelomate, there's no separation between the endoderm and the mesoderm. Okay? They lack a body cavity. Questions on coelomate, pseudocoelomate, acoelomate? All right? So coelomates and pseudocoelomates belong to the same grade. A grade is a group whose members share some characteristics, but they are not necessarily a clade, an ancestor of all of its descendants. So that's really, this is really getting to the point where you're discerning between a group and a clade. Okay? I'm not really that concerned in whether or not you know what a group or a clade is, but protostome versus deuterostome. What is the difference?
Protostome develops from the mouth, deuterostome develops from the anus.
All right, so it's really talking about where gastrulation is going to occur and what's the ultimate fate of that gastrulation in the beginning of gastrulation, whether or not it's going to be the mouth or the anus. So, also with this is a determination, a difference in how the cells divide, okay, during early embryonic development. Right? We know cleavage is fast, rapid mitotic divisions, the beginning of right after the zygote, okay? But in protostome development, cleavage is what we call spiral and determinate, okay? So spiral, really we're talking about where the axis of separation is going to be. Okay? So if I said that here's your zygote. Okay? And then if I'm going to determine that the axis, this zygote is going to split into two here. Okay? Now because this is determinate, it is now going to be roughly at 90 degrees to the first separation. So where is the axis of that first separation is in blue. Where is the next axis going to be? Okay, sort of that way. It is determinate, meaning it is going to have a certain destination. It's going to be sort of assumed that occurs somewhere. Okay? Whereby deuterostomes, cleavage is radial and indeterminate. And you'll see that in a second in the next slide. So in indeterminate cleavage, each cell in the early stages of the embryo cleavage retains the capacity to develop into a complete embryo. Indeterminate cleavage makes possible identical twins and embryonic stem cells. All right? So determinate meaning that they are sort of determined one way or another and where it's going to be. So here's protostome versus deuterostome. So spiral and indeterminate. Okay? Meaning that they're going to become certain things. Spiral. Look at the difference, the way they are. What do you see the difference between those first two parts? Radial versus spiral. The lower portion of these is a little sort of turned, right? So meaning that the axis for the has occurred, 1, 2, but now the next axis is sort of off center, whereby this is on center, sort of similar axes, right? They're radial, there's no off center turn that occurs. All right, here's your coelom formation is different. Okay, the beginning of there's the blastopore, the coelom in protostomes, right? Begins to form around the blastopore, whereby the coelom begins to form opposite the blastopore in deuterostomes. Right? And then finally you end up with the fate of the blastopore itself. Right? In protostomes, the blastopore will eventually form the mouth, and in deuterostomes, the blastopore actually will eventually form the anus. Questions on that? And the formation of gastrulation, or gastrulation in general, is the beginning of the formation of the digestive system. All right, so coelom formation, archenteron, sort of went over all this. I'll leave this. All right, the blastopore, the fate of the blastopore, photostome, what it ends. All right. And then we get to the last part of the chapter. So with radiosymmetry, we've done cleavage, we've done protostome development. All right. Now we get to the point where what the hell's the difference in terms of characteristics? All right. Why do we go through all these developmental traits? Really, it's about characterizing what is closely related to one versus not related to another. All right? A way of classifying animals and grouping them together according to their developmental traits. The more closely an animal is related to another, it should resemble or share quite a few traits, right? So with this is the idea of phylogenies, right? Phylogenies is a way of classifying things and separating them apart. So sort of what you did with your dichotomous key is a rudimentary phylogeny. It's sort of a classification, okay? Phylogenies now, that kingdom, phylum, class, order separation, combine your morphological, molecular, and fossil data. Prior to, I don't know, 40 years ago, everything was based upon morphological and fossil data that separated. But now with the emergence of technology, we can actually look at DNA sequences and compare them that way. Okay? So, couple different ways we can classify animals, right? Based on mainly morphological and developmental comparisons, what we've done in the past, versus what we do now, where we incorporate molecular data, all right? And it's not to say one is correct over the other, it's just a different way of comparison, okay? So what do you end up with? Here's your ancestral colonial flagellate, right? Your metazoa, then to eumetazoa. Eventually, you can break it down into bilaterians and their deuterostomes and protostomes. You'll look, you have sponges, jellies, you had some ctenophores, and then your deuterostomes, your brachiopodas, your starfish, your chordates. Okay? And then you have your platyhelminthes, rotiferas, mollusks, annelids, arthropods, and nematodes, right? Your protostomes. And they're separated based upon them. Okay? This is according to what? This is a separation according to their developmental and morphological characteristics. Right? Deuterostomes are together, protostomes are together, bilaterians are sort of together, and then you have all the radiosymmetries or asymmetrical animals up top. All right? Morphological structural data. Next. You still have sponges, your asymmetrical sort of animals up top. They're kind of arranged in a different way. Your protostomes and deuterostomes are mainly the same, right? Your deuterostomes are, you see along this branch, right here, right? But instead, your protostomes, according to molecular data, are actually split into two more, right? So these are they're not necessarily protostomes, right? But they're lophotrochozoans, right? Which include the platyhelminthes, your brachiopods, your mollusks, your earthworms. Why you have the ecdysozoans, include your arthropods and the nematodes. Right? And one of the defining characteristics of the ecdysozoans is that they undergo a process called ecdysis. Right? What do arthropods, mainly insects, lobsters, crabs, what do they do? What do they have? a hard exoskeleton. So how do they grow? How do they get bigger? They what? They molt. Molting is a process called ecdysis. That is a characteristic, right, that is unique sort of to arthropods. So they actually break their hard exoskeleton and move out from there. Okay? Ecdysis. What do they agree upon? All animals share a common ancestor. Sponges are the basic animals. They're part of the beginning, the link between single cell organisms to multicellular organisms. Right? Your eumetozoans is a clade of animals with true tissues. Right? And most animal phyla belong to are bilaterians. All right? And then you have your chordates, and some other phyla belong to deuterostomes. All right? So you have chordates and others. So that sort of separates protostomes from deuterostomes, regardless of what basis you're looking about. Okay? And then you have the process of sort of resolving the difference between the two. All right? So because the morphology tree divides bilaterians into two clades, your protostomes and deuterostomes. Okay? The molecular studies indicate 3 bilaterian clades: your deuterostomes, the ecdysozoans, and the lophotrochozoans. Right? Ecdysozoans undergo ecdysis. Okay, that includes the nematodes and the arthropods. All right, there's an example of a what is it called? Can't think of the name of it right now, but whatever. Undergo molting that's occurred. Right? That's not the word I was looking for. I was trying to name. That is a bug. And your lophotrochozoans have what's called a lophophore, a feeding structure, and then others go through this drink called the trochophore larva stage. All right, but so whether you talk about structural or molecular phylogeny, there's some similarities, and then sort of they get into a point where the protostomes are divided into 2 other clades in molecular data versus the structural data. All right, and here's your lophophore. All right, looking at the structure and then the structure of a trochophore larva, depending upon the stage you're looking at. Where do we go from here? As we get larger databases of structures, we can compare RNA structures, DNA sequence data, and come up with a better characteristics. All right, better characteristics and better separation between animals classifications. Questions on chapter 27? No? Okay. We're done.